Hi guys, let us begin with the same topic what we had the last class. The laws of Mendel's, the Mendelian inheritance. So based on the monohybrid cross what is seen in the last class, Mendel proposed two basic principles. Now these basic principles called as law of dominance or law of segregation. So the name is the laws, that is named the first law or law of dominance, the second law or law of segregation. And these two results came only after the experimental analysis of monohybrid cross. So what are the basic concepts, basic principles according to the law of dominance? As Mendel said, the characters are controlled by some stable, discrete physical units. The physical basis of unit, the physical basis of heredity. So what are called as the factors. So these factors are responsible for controlling a particular character in an individual. So whatever may be the number of individuals, we have a particular character being controlled by a particular gene or a factor located at a particular locus in all the individuals, irrespective of the number of individuals. So, number one, the characters are controlled by discrete physical units called the factors or genes. Mendel called this one as factors. Later Johansen called this one in 1903 as genes. So the factors always occur in pairs. Suppose you take homologous chromosomes. See that one. There are two genes, there are two copies for a particular character, and these two genes are located in homologous chromosomes. And here are represented genes, the factors, dominant factors, or simply the capital letters E. They are being localized at the same point on the homologous chromosomes. So for every character, we have two factors or genes. They are localized on the homologous chromosomes. That is the second one. Factors always occur in pairs, responsible for controlling a particular trait. Now in a dissimilar pair of factors, for example, in some cases, the homologous chromosomes may not have the same factors. In one chromosome, we have a capital letter. In another chromosome, we have a small letter of the same alphabet. And these two alphabets are actually the alleles controlling a particular character. So when these two alleles are present, these are called the dissimilar pair of alleles. So in this case, the dissimilar pair of factors, one factor is dominant over another. For example, the factor E is always dominant over other factor, namely the small alphabet, small e. And that is being considered as a recessive factor. So as the monohybrid cross he conducted, so in a dissimilar pair of factors where we had capital T and small t, so one member, namely this capital T, is dominant over the other. Now this capital T is called as a dominant factor and the small t is called as a recessive factor. So these were the actual observations made by him based on the monohybrid cross. So the factors always occur in pairs, they control the characters, and normally in a pair of dissimilar factors, one factor is always dominant over other factor. Then, so these are all the basic principles in monohybrid cross observations. Now what is the use of this law of dominance? Why we are using this one law of dominance? So what do you mean by law of dominance? Suppose when a pair of pure line contrasting characters plans are crossed, only one of the two characters being expressed in F1 generation. So in that case what is seen already, that is a tall character. Another character being suppressed. So the principle of law of dominance, this is the principle, and that is why I am using, that is the use of law of dominance. What is its main use? To explain the expression of only one of the characters in F1 generation and expression of both the characters in F2. So the first character, the tall character, is expressed in the F1 and both the characters, namely the tall and recessive, being expressed in F2. So that is a use to show that only one character being expressed in F1 generation and both the characters being expressed in F2 generation as per the monohybrid cross is concerned. Now another use of this dominance, so we are obtaining the proportion of tall and dwarf in a monohybrid cross 3:1 to explain the proportion, how far this 3:1 obtained in the monohybrid cross. These are the two main uses of law of dominance when two pure breeding varieties are crossed for a monohybrid cross. One, that is the expression of only one character in F1 and the expression of both in F2, and this one could be explained only with the help of monohybrid cross according to the law of dominance. Another one, we are obtaining the ratio 3:1 in a monohybrid cross in F2 generation. How far we are getting this ratio, and that is being explained only based on the law of dominance. Now, the second law, that is law of segregation. So this is possible only when the two genes in a hybrid or the two contrasting factors in a hybrid so do not just actually blend. The word blend refers to the mixing or interaction. So this law is based on the fact that the alleles do not show any blending at the F1 stage. This is possible only when the two genes, for example, in the homologous chromosomes, we have capital T and small t, and this is a hybrid or a heterozygous F1 having both the alleles. If these two alleles do not mix with one another, then only the law of segregation is possible. So, this law is based on the fact that the alleles do not show any blending at the F1 stage. If there is any blending, we cannot get back the recessive character once again in the F2. The reappearance of F2, the reappearance of the F2 recessive character or the reappearance of the recessive character in the F2 clearly indicated that the two genes for tall character and the recessive character did not blend. That is why we have the reappearance of the recessive character once again. So the law of segregation is possible only when the genes are not blending in the F1 generation. Now, in the case of law of segregation, now this is the parent, let us assume, having the genes capital T, small t on homologous chromosomes. When these parents produce gametes by the process of meiosis, what will happen? The diploid number of chromosomes being reduced to haploid. The diploid number of chromosomes being reduced to haploid. Same is the case here also. Out of the two genes, one enters into a particular gamete for a particular character, another one enters into another gamete. So this is what we call this one segregation. So when the gametes are formed during meiosis by the parents, the two alleles for a particular character segregate or separate from each other, and they enter into two different gametes. As a result, each gamete receives either capital T or small t, either of these two alleles only and not both. This is because of the segregation or separation of the two alleles during the formation of gametes. So when the parents produce gametes by the process of meiosis, the factors or alleles of a pair, so here the factors or alleles of a pair, capital T and small t, separate or segregate from each other. That is the law, that is law of segregation. You'll see how far it's taking place. So as a result of the segregation, what I mentioned earlier, so we have, this is F1 hybrid, and this one forms a gamete. So we have received two gametes, one with capital T, another one with a small t. So each gamete receives only one of the two factors, either capital T or small t. So this process of segregation is a natural process, a random process, and so there is 50% chance of each gamete receiving either allele. So each gamete receives only one of the two alleles and not both. So there is only 50% chance of obtaining either of the two alleles. Now, normally, so I am concerned with this heterozygous pair. The two alleles are different, what I mentioned here. Suppose, let us take, I am taking a homozygous individual. This is a homozygous, capital T and small t, the homozygous individuals I am taking. So when the homozygous individual form the gametes, so once again we are receiving the gametes with the haploid number of chromosomes, one with half the number of chromosomes, another one with half the number of chromosomes of the parental varieties. Here I am taking two letters for homozygous condition. So when the homozygous individuals produce gametes, all the 100% gametes are similar. and they are having the same what is called allele for that character. But when the heterozygous individuals form gametes, suppose this is a heterozygous individual, it produces 50% of the gametes with a capital T and 50% of the gametes with a small t. Suppose we are taking the heterozygous, when it forms gamete, 50% of the gametes with a capital T and 50% of the gametes with a small t. So that is happening in the case of heterozygous individuals, but not so in the case of homozygous, because they have similar alleles. So all the gametes are similar in the case of homozygous individuals when they form the gametes by meiosis. But in the case of heterozygous individuals, 50% of the gametes with a capital T and 50% of the gametes with a small t. So here you see that one. So this segregation of allele is a random process. What I mentioned is in a natural process. And so there is a 50% chance of a gamete containing either allele, either capital T or small t. Then homozygous parent produces all the gametes similar, what I mentioned earlier, while the heterozygous one produces two kinds of gametes, 50% with capital T and 50% with small t in equal proportion. So we have two kinds of gametes, each having only one allele, either capital T or small t, that is in equal proportion, what I said, that is 50 and 50. Now, we have received the fertilization process. There is a probability. So each gamete has a chance of fertilizing. Suppose for example the sperm. A sperm with a capital T has a chance of fertilizing the egg with capital T. And similarly, it also has equal chance of fertilizing an egg with a small t. So it is a random process, a natural process. So as a result of this one, what is called the random fertilization, we have received the resultant zygotes. And these zygotes have the genotypes of capital T, small t, and small t, small t. So all the three types of genotypes will be produced when fertilization occurred by means of random process. Because of the probability we have, the genotypic ratio 1:2:1. That means the capital T has a chance of fertilizing capital T, so you are getting its homozygous, that is dominant, and the capital T also has a chance of fertilizing an egg with small t. So we have received the heterozygous individual. And similarly, for example, when a recessive gene, that is a gamete having a recessive factor, fertilizes an egg having a recessive factor, will have received what is called the recessive homozygous individuals. Then, so as a result, we are getting actually three genotypes. One homozygous dominant, another one heterozygous dominant, another one homozygous recessive in the ratio of 1:2:1. That means 1:4, 1/2 and 1:4 ratio of this capital T, then heterozygous dominant and homozygous recessive. We can express this one in the form of mathematical, actually binomial expression. That's ax plus by whole square is equal to, so 1:2:1. So that means each gamete is normally having the genes capital T or small t in equal frequency of 50%, that is 1/2. Now, the expression is expanded as given below. See, 50% capital T and 50% small t. So, each one has the probability of fertilizing another gamete, that is opposite gamete. That means either egg or in the case of sperm, as the case may be. We have received the same expansion, the binomial expression, 1/4 capital T plus 1/2 capital T small t, and then 1/4 just small t. That means 25% homozygous dominant, 50% heterozygous dominant, and 25% homozygous recessive. So this is actually what we have in respect to the law of segregation. So in the case of law of segregation, whatever number of characters we are taking while crossing the individual, you know that an individual is nothing but a collection of many characters. So we cannot isolate the gene, and also we cannot isolate the characters. So only thing we can do, we can concentrate only on a particular character and also a particular factor. how far the characters have been segregated, how far the genes have been segregated only for that particular character. So as a whole, we cannot concentrate how many characters or how many just possible actually the factors what we have being inherited or being separated or segregated at a single time. Though all the factors are being inherited at the same time during fertilization, we can concentrate only on a particular factor's inheritance, particular factor segregation, how far the factor has been transmitted to the next generation, namely the progeny. Now, so based on the monohybrid cross also, Mendel has found certain things. So we are making a cross between one parent with another parent. We do not know the nature of the dominant parent. So there are two types of dominant parents. One is a homozygous dominant, another one a heterozygous dominant. So these two parents are not externally visible and both are externally similar. So we cannot know or we cannot just observe and find out the nature of the genotype of these two individuals as both are exactly similar. For example, if you take a homozygous Tom and heterozygous Tom, and though they are actually genotypically different in their genetic constitution, externally both are alike, only a slight variation. So, if you want to know the nature of the dominant parent who is taking part in a cross, we have to make a particular cross. Whether the dominant parent is homozygous or heterozygous, to find out the nature of the parent, we have a specific cross. The name of the cross is called as a test cross. So, normally it is also called as a back cross. So, we are taking in this back cross the F1 hybrid. So, for example, this one F1 hybrid, a heterozygous F1 hybrid, is being crossed with either with the dominant parent or in some cases with the recessive parent. And both are considered as so we are making a cross. This is F1 heterozygous individual, capital T, small t, and that is being back crossed or crossed with either with a homozygous parent or with a homozygous recessive parent, homozygous dominant parent or with a homozygous recessive parent. As we are going back to the previous generation for just selecting the individual for the cross, this is also called as a back cross generally. Now the first one, crossing of this F1 individual with homozygous dominant, this is called dominant back cross. We are back crossing the F1 hybrid or heterozygous individual with the dominant parent, dominant homozygous parent. Or in another case, if you are making a cross of this F1 heterozygous with a homozygous recessive parent, then it is called a recessive back cross. A recessive back cross is a one in which we are crossing F1 heterozygous with a homozygous recessive parent. Now out of these two crosses, the recessive back cross is of importance genetically or just in base of genetics. In the basis of genetics, or we can have this one for analyzing the genotype of the parents. So normally the cross between a heterozygous parent and a homozygous recessive is called as a test cross. Because it is a supporting cross, this cross is normally used to find out the nature of the dominant parent involved in the cross. So, if you want to know the genetic nature of the parent, particularly the dominant parent, we have to take the unknown genetic parent, and that unknown genetic parent is being crossed with a homozygous known recessive parent. This cross is called as a test cross, what I represented earlier. So a cross between an organism with an unknown genotype. We do not know the genotype of that dominant parent. So normally we are taking the dominant phenotype, either dominant homozygous or dominant heterozygous. So this is an unknown genotype parent. The cross's aim is to find out the nature of the dominant parent, whether it is homozygous dominant or heterozygous dominant. So here it is being crossed with actually a known recessive phenotype. Here we are crossing a heterozygous or a homozygous top with a recessive parent. You are crossing either this parent or this parent with this one. So then only we can get the idea whether the dominant parent is heterozygous or just homozygous. And that is the main aim of this class. So that is always called as a supporting class, to find out or to distinguish the nature of the dominant parent as heterozygous or homozygous. Now, I mentioned earlier, it is a supporting class. The main aim of this class is to distinguish between the heterozygous dominant and the homozygous dominant. Suppose, if the dominant parent is heterozygous, say an example, capital T, small t. This parent we are crossing with a recessive parent. In such crosses, we obtain individuals in the F2, for example, tall and dwarf in equal proportion. See that one, in this case we are making a cross between a heterozygous dominant and a homozygous recessive. If the dominant parent is heterozygous, then we are getting actually this tall and dwarf parent in a ratio of 1:1. So in the case of test cross, if in a hybrid cross, the phenotypic ratio of F2 is 1:1, then the genetic nature of the parents will be heterozygous dominant and homozygous recessive. I repeat once again. If in a hybrid cross, the phenotypic ratio of F2 has 1:1, then the genetic nature of the parents will be heterozygous dominant and homozygous recessive. Suppose if the unknown parent is having a dominant genotype when crossed with a recessive parent, we'll get 100% of the individuals all dominant, then it is called actually a homozygous dominant parent. So if in a hybrid cross, if you are getting all the individuals in that progeny similar or 100%, then we can say the dominant parent involved is homozygous dominant. So, if the dominant parent is heterozygous in a test cross, you will get the phenotypic ratio of 1:1. If the dominant parent is homozygous, you won't get this phenotypic ratio of 1:1, and all the individuals are similar, having the same phenotype. So, this is actually used to distinguish the difference between a homozygous dominant and heterozygous dominant parent. So here is given the diagrammatic representation of test cross. So on the left side we have received all the individuals similar. So in the case of garden pea plant, regarding the flower color, one is violet, another one is white. So normally the violet color of the flower is dominant over the white. Now in the left side we are crossing the unknown parent. That is why here given the question mark, the unknown parent, that is the parent without known genotype. That is why it's called as unknown parent. So a parent with unknown genotype, we can say. So a parent with unknown genotype is crossed with homozygous recessive. Now we received all the individuals are similar, 100% violet. I mentioned earlier in the case of tall, that is the previous illustration. Here we are taking the flower color. If all the flowers are violet, then we can say that the dominant parent involved in that cross is nothing but actually homozygous dominant. Now on the right side we have a different result. So we are taking once again this violet flower. It's being crossed with what is called homozygous recessive. The same cross. So these two are actually, this is an unknown parent with unknown genotype. But here the expected result is different from that of the left side. Here we are getting half of the flowers with the pink color, or we can say what is called the violet color, and half of the flowers are white. So if we are getting such a ratio of 50-50, then the unknown parent is heterozygous. That's why I mentioned earlier with the illustration with the tall character and dwarf character. Here we are taking another character, namely the flower color. So in a hybrid cross, if you are getting the ratio of 1:1, as mentioned the right side regarding the flower color, definitely the parent would be just the dominant parent would be, that is unknown flower is here is heterozygous, or the unknown parent is heterozygous. On the left side, we have all the parents are similar. If you are getting such a result, definitely the unknown parent is homozygous dominant. So this is actually used in genetical principles because we know only the external appearance of an individual, not the internal organization of the genes. And for that we have to make such crosses because the genes are not visible. We cannot see the genes internally. We cannot just see the genetic makeup. Only by mix of test cross, you can just understand what is the genetic constitution of that particular character. Now, so let us take up. So how far the law of dominance is working? What is the principle lying behind it? So the explanation for the concept of dominance. Now, you see that one. So each and every character is controlled by a particular gene. Indirectly we can see, in another words, each gene contains information for a particular trait. Each gene is coded. Each gene has a blueprint for the expression of a particular trait. Now, as we are all, say an example of human beings, diploid organisms or any other organisms. And for each gene there are two copies. There are two copies. And these two copies are called as alleles. So a pair of alleles. So for each gene there are two copies and they are represented as a pair of alleles. So normally these two alleles may be similar for a particular character or need not be similar always, as in the case of heterozygote. So for example, I mentioned here, so I am taking capital W, small w, and again here capital W, capital W. So you can see this one, the statement for this one, these two alleles need not always be identical. In a homozygous organisms, we have one dominant allele, one recessive, in a heterozygous organism, we have one dominant allele and one recessive allele. But in the case of these homozygous organisms, we have the two alleles or the two copies are identical. But it is not identical in the case of heterozygous organisms. Now, what is the reason for that one? Normally, the character, a normal character is called as a wild type. A normal character is called as a wild type. So because of certain changes, what we call this one, mutation. Because of mutation, so what will happen? The dominant allele may get changed. So when the allele gets changed, what will happen? The character can also be changed, or the coded information that that allele contains may also be changed or may also get affected. So in diploid organism, as I stated, there are two copies of each gene as a pair of alleles. These two alleles need not always be identical, as I mentioned here in the heterozygous organisms. Now, see that one I mentioned, just suppose I am taking capital W and small w for the flower color violet. So one of them may be different. You see that one, this is a different allele. This is a different allele. Due to some changes that it has undergone. This is because of certain changes, because of certain factors, or because of mutation. And as a result, the information originally contained in the normal allele has been changed here, because it undergone mutation. So the information is mainly concerned with any biosynthetic pathway or formation of any product inside the living cells, an example of hormone or enzymes, etc. So because of mutation, one of the two has undergone change. While it gets changed, it normally affects the information that it contains. So it can be expressed. It can be expressed in a different form. So a normal allele can express its own character. When it undergoes changes, it expresses some other character, or the character may not be expressed, or a defective character is formed. Even in some cases, a normal character is formed, as in the case of a normal allele. So anyway, let's see with an example. Now, so I am taking a particular gene. Say an example of gene A that contains an information for the synthesis of a specific enzyme. Say an example of E. This is the enzyme. And this enzyme is responsible for the transformation of a substrate S. So this is a normal allele and this is a normal product because of the information contained in the gene, and this product is formed, namely the enzyme, a protein, and that enzyme is responsible for the transformation of this substrate S into S1. Same example. This is actually the action performed by this enzyme E. But suppose if this allele A has been changed. So this is number one, a dominant allele, a normal allele we can see produces an enzyme, and this enzyme is responsible for the conversion of one substrate, for the transformation of one substrate into another. Suppose this allele gets changed because of mutation, it gets modified. Now this allele may produce a normal enzyme, or we can have less efficient enzyme This is one. So when the allele undergone change, for example, it's been converted to small a, it may produce a normal enzyme just like this allele, or it produces less efficient enzyme, or sometimes it may not produce a normal enzyme, or it produces a defective enzyme. So there are three possibilities. So a gene that contains information for producing an enzyme, a normal allele produces a normal enzyme that is needed for the transformation of a substrate S, what I represented. So the modified allele here I mentioned, for example, small a, could be responsible for the production of three possibilities I mentioned. One, either it produces a normal enzyme or less efficient enzyme, or it produces a non-functional enzyme, the one which is not working properly, or it has no ability, or it may not produce an enzyme at all. So what is the ultimate effect? Now in the first case, when this modified allele produces an enzyme similar to the enzyme produced by the normal allele, and also similar to the normal allele in the expression of the phenotype. So that is actually, so in the first case, the enzyme being produced by the modified allele. So in this action it is similar to the normal allele, the expression of the phenotype. And such equivalent alleles are very common normally in nature. Suppose when the allele produces non-functional enzyme or no enzyme at all, what is called this modified allele, then the phenotypic expression of the character is affected. No normal phenotype will be produced. Normally, the original phenotype or the normal phenotype depended mainly on, that is what is called the unmodified allele. The unmodified allele or the normal allele is responsible for the production of normal enzyme. That's why I said. So the production of normal enzyme depended mainly on or only on unmodified allele. As here the allele has been modified, no normal enzyme being produced. So in such cases, the modified allele is considered as the recessive allele, and the unmodified allele is considered as what is called the dominant allele. So I mentioned earlier, so in the first case, when the modified allele produces enzyme similar to the effect of a normal allele, namely the unmodified allele, so they are equal in their expression. So such equivalent allele pairs are very common. If the allele produces non-functional enzyme or no enzyme, the phenotype may be affected. The phenotype will only depend on the functioning of the unmodified allele. So it purely depends on the functioning of unmodified allele. Already just instructed and just I said you. So the unmodified allele, which represents the original phenotype, what I said earlier, is the dominant allele. An allele without undergoing any change, The allele without being affected is called as a dominant allele. And the allele which is being affected, the allele which is being modified is considered as a recessive allele. So, anyway, the law of dominance is based on this fact only. So, when some change is taking place, one particular gene has been modified. When that gene has been modified, ultimately its expression, the information that contained, also modified. So, it affects the expression of the character. So, normally the modified allele is called as a recessive allele. The unmodified allele is called as a dominant allele. So, these are all the basic principles what we have to follow in the case of law of dominance. So, in a dissimilar pair of factors, one factor is dominant over other factor. The dominant factor is always the normal allele. The recessive factor is normally formed because of certain changes. For example, in the case of Drosophila, the eye color is red or white. So, the red eye color is dominant over white eye color. So, here the red eye color gene is called a modified allele, a dominant allele. The white eye color allele or the gene is a modified allele or a recessive allele. So, likewise, we have so many examples in genetics. Now, after the principle of law of dominance, we have to also differentiate this law of dominance from some other processes, namely incomplete dominance and then codominance. So, we have complete dominance, incomplete dominance and codominance. Now, what do you mean by incomplete dominance? Suppose let us take two factors. Say an example of a R and then small r. These are all the two factors controlling some characters in organisms. So these are all some of the factors, contrasting factors called as alleles, responsible for the expression of character. So when cross is affected between two such individuals, normally according to the law of dominance, the F1 resembles either of these two parents. Either of these two parents will get either R parent or small r parent. This is what is called law of dominance. But in the case of incomplete dominance, when a cross is affected between two contrasting pure line varieties, we have received F1 individual. That F1 individual is in between, intermediate between the two parents which are involved in the cross. So, such a result has given the idea of what is called incomplete dominance. So, as per the law of dominance, as per the law of segregation, the genes do not blend. There is no mixing of genes in F1. But for this incomplete dominance, it is based on the principle of blending. In law of dominance, there is no blending, and also in the case of law of segregation, that is possible only when the two factors in a hybrid do not blend. But in the case of incomplete dominance, we are getting a new result in F1. That character is intermediate or in between the two parents. That is possible only when there is a blending of the two factors. The interaction of the two factors that produce entirely a different character. Now, what is incomplete dominance? So, incomplete dominance is also called as blending inheritance because we have the blending of the two factors. It is also called partial dominance because we are getting entirely a different character, not a complete one. A complete dominance is not possible in the case of this one. So, in incomplete dominance, for example, if I am taking a hybrid, for example, I am taking this capital or smaller. These are all the two alleles. The two alleles of a heterozygote. So, the two alleles in a heterozygote influence the expression of a particular phenotype. Both together interact, both together influence the formation of an intermediate character. That is called incomplete dominance. So, incomplete dominance, the two alleles of a heterozygote show influence the expression of a particular character. So, both are taking part, both are blending together to form a particular character. That is called incomplete dominance. So, the condition in which both alleles of the heterozygote Influencer phenotype is called incomplete dominance, and both are taking part, blending together to produce a particular character, and that is called partial dominance or blending inheritance or incomplete dominance. Now let's see with the example. So there are two examples. In plants and one example in the case of animals. In the case of plants with reference to flower color, one plant, just what is called the snapdragon, looking like a dog in sitting posture, called as a dog flower. The binomial Antirrhinum majus, and another one, the four-club plant, Mirabilis jalapa. So in these two cases, we are taking the flower color. So there are two contrasting flower colors. One is red, another one is white. When a pure line red variety is crossed with a pure line white variety, then what was observed by coherence? It was observed by coherence. That inheritance was observed by coherence. That is, in these two plants, the F1 they received. So he received a pink variety, an intermediate character between these two, with a genotype like this, capital or small r. Heterozygous individual, but we have the expression of the character, that is intermediate character, what we call this one as blue color. So red and white, we are getting this one. When the F1 individuals are self-fertilized, again we received the plants. Interestingly, when compared to just actually the normal monohybrid cross, there you know that one, the genotypic ratio of monohybrid cross is 1:2:1. Here also we are getting the genotypic ratio as in monohybrid cross 1:2:1, but the phenotypic ratio of 3:1 in the monohybrid cross has been modified. From 3:1 has been changed from 3:1 to 1:2:1. So I am saying the meaning for that one. Both the phenotypic ratio and the genotypic ratio would be the same in the case of incomplete dominance. In F1, an intermediate character, no parental character being expressed, that is why it's called incomplete dominance. The F2, we are receiving the parental characters along with intermediate characters in the ratio of 1:2:1. So this is what we have, the phenotypic ratio. That phenotypic ratio is equal to the genotypic ratio in the case of this incomplete dominance. But in the case of monohybrid cross, a normal dominance cross, the phenotypic ratio is 3:1 and the genotypic ratio is 1:2:1. But here both the phenotypic ratio and the genotypic ratio would be the same. We'll see the illustration. So I made a cross. I mentioned the cross is that one, he made a cross. A cross between two breeding red flowers and the two breeding white flower plants. So the F1 was pink. Then it appears neither the red nor white color trait is dominant, and both are actually equally just dominant. That is, they're interacting together and producing a character. Neither red nor white is considered as a dominant. But when F1 self-fertilized or self-pollinated, we receive the F2, result in the ratio of red, pink and white. Now this is the genotypic ratio as well as the phenotypic ratio. So given inside the brackets, the genotypes, and then you have the phenotypes of red, pink and white. So both phenotypic ratio and genotypic ratio would be similar. So in complete dominance, we have the phenotypic ratio 3:1 and the genotypic ratio 1:2:1. But in incomplete dominance, both the phenotypic and genotypic ratios would be the same. Let's see the illustration. Now, I mentioned earlier, just the phenotypic ratio 3:1 has been changed to 1:2:1, but the genotypic ratio is similar to that of complete dominance. So here is the example. So when a dog flower, so when a plant, for example, Antirrhinum majus, normally called as the dog flower plant, so the red flower is crossed with the white, we received all pink. So an intermediate character between these two. When this pink flower was self-fertilized or self-pollinated, we received the F2 generation having the phenotypic ratio of red 1, pink 2 and white 1. And also having the genotypic ratio of 1 homozygous dominant and 2 heterozygous dominant of intermediate and 1 that is homozygous recessive. So as genotype wise, Homozygous dominant, heterozygous dominant, and homozygous dominant in the ratio 1:2:1 as in the case of monohybrid cross. But the phenotypic ratio has been modified from 3:1 to 1:2:1 here. Now, so another type of dominance, one is complete dominance, another one incomplete dominance. Now let's have codominance. Now for example, if we are taking two parents which are pure. So when we are crossing these two pure parental varieties, we have received the parent with both the characters present side by side. For example, in the case of cat, you have watched a cow or just actually a bull having patches of red and white. Or in the case of puppies, you can have patches of black and white. They never mix. And both the characters being found side by side. This is because both the factors are equally dominant. They are showing individual expression. That is why we have in some cattle patches of white and patches of red together, but there is no mixed color. Even in the case of puppies, we have patches of black and patches of white, and they never mix it. We have just two different characters but presented together. So they are working together, presented together. They are expressing their individuality. Now here in codominance, again it is a condition. For example, I am taking the blood group AB blood group in man. AB blood group. It is the best example for codominance. Now in the case of AB blood group, we have both the genes A and B. This is a hybrid or a heterozygous. AB blood group is an example for heterozygous condition. Now we have both A and B genes. They are presented together, but they never blend, as in the case of incomplete dominance. They never blend, but they are presented together, and so that they express their own phenotype. The A gene produces A antigen, B produces B antigen, and both being present in the same person in the obese. So, if you consider this one AB as a heterozygous, the two alleles in the heterozygote show individual expression of each allele in the phenotype. So, individual expression of each allele in the phenotype. For example, if you are taking, I am representing a simple way of the blood group. This is a sample of blood group. The small circles represent the RBCs. Now, in the case of AB blood group persons, we have some of the RBCs have A antigen, some of the RBCs have B antigen. Some of the RBCs have A antigen, some of the RBCs have B antigen. So, you see that one, there is no mixing of A and B antigens, and both are present as such together, separate. There is no mix. So, I will represent once again. We will come back later. Now, codominance is the condition where the two alleles in a heterozygote show individual expression of each allele in the phenotype. That's the concept what we have. I mentioned already the best example, AB blood type. So if you want to know the basic principle of this codominance, you have to go for what is called a phenomenon of multiple alleles. Because the blood group is the best example for multiple alleles. So multiple factor is different from multiple allele. We have multiple factor hypothesis. For example, the skin color. The multiple factor hypothesis is also called polygenic inheritance. Now, in the case of this ABO blood group, a single gene being represented by more than one form, then it is called multiple alleles. What do you mean by multiple alleles? Let us take gene A. This is gene A. So normally we have only two alternative alleles, either capital E and small e. So these are the two alleles, A and E, for contrasting characters. An example of capital T and small t are alleles. Capital E and small a are alleles. And similarly in the case of red color, for example white color, capital R is an allele to small r. So like that. So for every character there are two contrasting varieties or two contrasting characters. But that means we have only two genes for the characters, capital E and small a alleles. But in some cases we have more than two alleles. For example, the A allele being represented by I am writing just you see that the sequence. A single allele being represented by more than one form. A single allele being represented by more than one form. So here I represented, you see that one just A, and then in just a small alphabets up to 4 there are 6. So when a gene is being represented by more than one form, then this gene is an example for multiple allele, and the phenomenon is called multiple allelism. So what is the meaning for that? What occurrence of more than two alleles? See, more than two alleles. All these alleles are affecting the same character, and all these alleles are located at the same locus in a chromosome, but affecting the same character in different individuals. And such alleles are called multiple alleles. I repeat once again, occurrence of more than two alleles affecting the same character, located at the same locus, an example of blood group allele. It is located in chromosome number 9. Then such allele is called multiple allele, and the phenomenon is called multiple allelism. The ABO blood group is the best example for multiple allelism. Now, if a gene has more than two alleles, these alleles are called multiple alleles. You have the example, that is multiple allele ABO blood group. It was first represented by, that is called Landsteiner in 1902. He was the first person to identify the blood group, but his identification was based on the biochemical analysis, based on the presence or absence of two antigens A and B. So biochemically he classified the blood groups, not genetically. So Paul Landsteiner just proposed the ABO blood system. So we have 4 types of blood groups. Generally we can see A, B, AB and O. And now how are these blood groups are formed? The blood groups are actually based on the genetic factors. Now, example for multiple allelism. Now let's see. And the basic principle, the basic factor, the gene responsible for the blood group was first proposed or represented by Bernstein in 1924. Bernstein in 1924 discovered that the ABO blood group normally controlled by a particular gene, namely I gene. This I stands for isoheme agglutinogen gene. Isoheme agglutinogen gene. And this gene is being expressed by more than one form. It exists in 3 allelic forms. For example, and before that, this gene I gene, isoheme agglutinogen gene, is responsible for the production of a particular sugar polymer. So the RBCs of all humans on their surface have a sugar polymer, a polysaccharide, and it is being projected over the surface of the membrane. Now normally we have 2 kinds of sugar, and these 2 kinds of sugars or polymers are controlled by actually the gene. The gene is normally formed in 3 allelic forms. 3 allelic forms. So the gene I has 3 alleles, namely A, B and I. Now in these 3 alleles, now is this IA gene and IB gene, you see that one the dominant letters, and both are dominant over this gene, namely the small I gene. So the alleles A and B produce a slightly different form of antigen, and both are dominant over this one. Now this IA gene produces the antigen A, a kind of sugar polymer, and IB gene produces antigen, what is called, that is AB antigen. So it is also a sugar polymer, whereas this recessive gene, the small letter, doesn't produce any sugar. That is why people having 2 genes of I have no antigens at all. They are normally considered as a O blood group. And people with A antigen are considered as an A blood group persons, and likewise B. If both A and B antigen is being present in an individual, then that blood group is called as AB. So the AB blood group is being represented because of the presence of 2 genes A and B in the same individual because of codominance. So as we have actually deployed, So normally we have two of the three alleles in our body for controlling the blood characters. So A and B are completely dominant over, that is I mentioned earlier. So it's being represented in the form of formula. IA is dominant or equally dominant to IB, whereas I is recessive to both. This is the actually representation. IA is equally dominant to IB, that is co-dominant to each other, we can say. So I is recessive to both. So when A and B are present together and both express their own sugar polymers, that is why that individual has that is AB antigens in their blood, considered as AB blood group. This is because of the codominance. Both are present together and expressing together individually. As we have actually three alleles, there are three different alleles. There are 6 possibilities of genotypes possible. There are 6 possibilities of genotypes. For A blood group, 2 genotypes. For B blood group, 2 genotypes. For O blood group, only 1 genotype. And for just AB blood group, 1 genotype. Together, 6 genotypes are possible. Now, so therefore a total of 6 different genotypes of the human ABO blood types and 4 phenotypes we have as mentioned here possible. And we can also, if any number of multiple alleles is given in the question paper, you can normally calculate the number of possible genotypes. So, by the formula, you see that one number of genotypes from multiple alleles is given by the expression n into n plus 1 divided by 2, where n is the number of multiple alleles. Here there are 3 multiple alleles, so 3 into 4 by 2, we can get 6. In the case of coat color in rabbits, there are 4 multiple alleles, so the possible number of genotypes would be just 10. So this is the way we can calculate easily how many possible genotypes would be normally occurred if you have a specific number of multiple alleles. So I have the table given. You just follow the table. So I compare that dominance, incomplete dominance and codominance. See that one, a very simple explanation. So in the case of dominance, F1 resemble either of the 2 parents I mentioned. In incomplete dominance, F1 was in between the 2 parents. See in the case of flower color. In codominance, the F1 generation resembles both the parents as in the case of AB blood group. So for further analysis, the possibilities of blood groups and other things I have represented in the table column. We go through the table column. If you have any doubt, ask me in the next class regarding this blood group. Here also the table given the possibility. Just let's suppose a parent has A allele, another parent has A allele. What would be the blood type in the case of offspring? Likewise, you can calculate based on the nature of allele derived from each parent. So you can get the result. Thank you. I will continue further in the next class.